Welcome to my next class. So we'll quickly look at what the mechanism of enzyme action is. Now, the basic mechanism of enzyme action is to catalyze the chemical reactions. Take note, which begins with the binding of the substrate with the active site of the enzyme. So there's a substrate. The enzyme, I told us about active site before, that the enzyme has an active site where it has to bind, where the substrate has to bind to. So on that enzyme active site now, the substrate has to bind to it. That is the beginning of what? Of enzyme reaction. Now, this active site is a specific area that combines with the substrates. Hello, I hope you are following me. This active site is a specific area that combines with the substrate. So we look at enzyme substrate interaction. Now, when there's that binding, when the substrate now binds to that enzyme active site, let's look at what happens. Enzymes are biocatalysts, which are high molecular weight proteinous compounds. It enhances the reactions which occur in the body during various life processes. It helps the substrate by providing the surface for the reaction to occur, that active site. The enzyme comprises hollow spaces occupying groups such as SH, your COOH, and others on the outer surface. The substrate, which has an opposite charge of the enzyme, fits into these spaces, just like a key fits into a lock. You know, I've said it before. Okay. Now, this substrate binding site is called the active site of an enzyme. The substrate binding site is called the active site of an enzyme. The favorable model of enzyme-substrate interaction is called the induced fit model. The favorable model of enzyme-substrate interaction is called the induced fit model. This model states that the interaction between the substrate and the enzyme is weak. Interaction between the substrate and the enzyme is weak. And these weak interactions induce conformational changes rapidly and strengthen binding and bring catalytic sites close enough to substrate bonds. Now, there are four possible major mechanisms of catalysis. We just quickly look at them. There are about four possible major mechanisms of catalysis. So quickly look at them. The first on my list here is catalysis by bond strain. Catalysis by bond strain. The induced structural arrangement in this type of catalysis, it produces strained substrate bonds that attain transition state more easily. The new conformation forces substrate atoms and catalytic groups like aspartate into conformations that strain substrate bonds. So we have covalent catalysis. The substrate is oriented to active place on the enzymes in such a manner that a covalent intermediate develops between the enzyme and the substrate in catalysis that occurs by covalent mechanisms. Best example of this involves proteolysis. Proteolysis, can check it out. Proteolysis by serine proteases that have both digestive enzymes and various enzymes of the blood clotting cascade. Now, these proteases, they possess an active site, serine, whose capital letter R group, hydroxyl, generates a covalent bond with a carbonyl carbon of a peptide bond and results in the hydrolysis of the peptide bond. So we've talked about catalysis by bond strain, covalent catalysis. Then the next one, the third one, is catalysis involving acids and bases. Other mechanisms add to the completion of catalytic events, which are launched by strain mechanisms such as usage of glutamate as a general acid catalyst. Catalysis by orientation and proximity, that is the fourth and the last here. So we have enzyme substrate interactions that induce reactive groups into proximity with one another. Also, groups like aspartate are chemically reactive and their proximity towards the substrate favors their environment in catalysis. So, action and nature of enzymes. We'll just look at it briefly and move to enzyme action. Once substrate binds to this active site in an enzyme now, they form a complex or intermediate, which then produces the product P and the enzyme E. I hope you're getting it. Once substrate S binds to the active site of the enzyme, they produce a product P and the enzyme E. Now the substrate which gets attached to the enzyme has a specific structure, and that can only fit in a particular enzyme. So every enzyme have their own active site. Hence, by providing a surface for the substrate, an enzyme slows down the activation energy of the reaction. The intermediate state where the substrate binds to the enzyme is called the transition state. By breaking and making the bonds, the substrate binds to the enzyme, they remain unchanged, which converts into the product and later splits into product and enzyme. Take note, and the free enzymes then bind to other substrates, and the catalytic cycle continues until the reaction is complete. So that is just it. So if you want to look at enzyme action, it's not more than what we have said, where we said enzymes have active sites where substrates we have to bind. Okay, so quickly look at what biological catalysts are, so that we can go to the next topic. Catalysts are the substances which play a significant role in the chemical reaction. Catalysts are the substances which play a significant role in the chemical reaction. Catalysis is the phenomenon by which the rate of a chemical reaction is altered. That action, the rate at which the reaction is altered or enhanced without change, that is catalysis. No change at all in this one. Now reaction takes place, and at the end of the day, nothing is altered, everything remains the same. That is catalysis. And we say catalysts are the substances which play a significant role in the chemical reaction. During a chemical reaction, a catalyst remains unchanged. I hope you are listening, both in terms of quantity and chemical properties. An enzyme is one such catalyst. We have so many catalysts, because somebody can say catalyst and enzyme, are they not the same? Okay, listen up. So an enzyme is one such catalyst which is commonly known as the biological catalyst. So enzymes present in the living organisms enhance the rate of reactions which take place within the body. And one thing you should know about biological catalysts, for example enzymes, they are what? Extremely specific. They are specific, that catalyze a single chemical reaction or some closely associated reactions. An enzyme's exact structure and its active site decide an enzyme's specificity. I've said it before. So every enzyme has their own active site, and any enzyme cannot just go and bind to any active site. No. So each active site has specific enzymes that can bind to it. So a substrate molecule attach themselves at the active site of an enzyme. Initially, substrates associate themselves by non-covalent interactions to the enzymes, which include ionic hydrogen bonds and hydrophobic interactions. Enzymes reduce the reactions and activation energy to progress towards equilibrium quicker than the reactions that are not catalyzed. So reactions that you have enzymes are faster, yes, compared to the one that enzyme is not there. So both eukaryotic and prokaryotic cells usually make use of allosteric regulation to respond to fluctuations in the state inside the cells. So we'll be stopping it at here. In my next class, we'll continue. We'll continue with our other topics. So I'll be looking at activation repression phenomenon. See you in my next class.